Dear audience, my name is Tamás Zsoldos. I am from the Institute of Experimental Medicine, and I would like to give you my presentation on the regulatory cells of our memories. First, I would like to start with a story. Meet Henry Molaison, a young factory worker who, from his 16th birthday on, he had epileptic seizures daily. His life was insufferable. His doctors could not find any medication that could help alleviate these seizures, so they decided to turn to surgery. On September 1st of 1953, they decided to remove a part of his brain here in the very center. They know that this was the center of his epileptic seizures, so they decided to remove it. The next day, Henry Molaison woke up and noticed that he did not have any seizures. He was feeling well, so he went back to his doctors, introduced himself, and said that he is much better now. So the next day, he went back, he introduced himself to his doctors, and said he was feeling well. The doctors noticed that why would he introduce himself? They already know who he is. But they realized that Henry Molaison could not generate any new memories, and that he forgot a lot of his previous ones too. So they realized that by removing the central part of his brain, they took the ability to generate new memories for him. So they realized that this funny structure here in the very center of our brain, the so-called hippocampus, is the center of our memory. It has quite a unique shape, as you see. It resembles somewhat seahorse, hence the name hippocampus, meaning horse-like sea monster in Greek. Since then, they recognized its functions not only in memory generation, but so learning emotional behavior and also localization. But in the brain, we cannot tell anything about its function and how it works simply by looking at it. We have to take a closer look. If we take a coronal section of the hippocampus, you can see, we cut it in half, we can see it has two main parts. The CA region, this is a horn-like structure, and the dentate gyrus in the middle. But this is still not enough. We can see it with our eyes. However, if we zoom in a little closer, we can see that it has layers to it. And the CA region has 5 layers, and it has a very broad layer, the so-called stratum pyramidale, that hosts the principal cells of the CA region, the so-called pyramidal cells. These are the main projecting cells found here. Separated by the hippocampal sulcus, we can see that there is the dentate gyrus with only 3 layers, and its principal cell layer has granule cells. They have distinct functions, however, it is important that they will be one of the main characters today. So, the principal cells receive sensory input from higher order cortical neurons, also from the entorhinal cortex found next to the hippocampus itself. Only a few % of the principal cells in the hippocampus encode events by firing together and generating a pattern. These are called engram cells. These are selected based on the network stimulation status and random characteristics. So if we are acquiring a new memory, a couple of neurons fire together, thus encoding one memory. But when we sleep, these engrams mature, they get transferred to the long-term memory. This is called memory consolidation. And when we activate these cells, the same subset of principal cells, so the same engram, can be reactivated during the process of memory retrieval. So we can remember what we have experienced at the moment of encoding. If we want to take an anatomical picture of how a memory looks like, here you can see the cells that were active during one single event. So basically, we can see how memories are stored. However, if we look more carefully, we can see that principal cells are not the only players here in the hippocampus. There are other neurons too. There are interneurons. These are mainly inhibitory GABAergic neurons, and there is a lot of them, not just in number, but also in variety. Here you can see that this article described 21 only in the CA region. I mean, that's a lot. And to show more variety, you can see that there are cells that are more localized to only one layers. These are mainly local inhibitory cells. However, there are more funny shaped, the inhibiting principal cells further away. And there are also interneurons that play a role more in local oscillations and how the hippocampus can work as a whole, generating a frame to encode memories. If we recall, to choose these engram cells, we have to inhibit these cells, not only just getting excitatory input from the cortex. And for this, we have interneurons. The principal cells receiving cortical inputs on dendritic segments are specifically inhibited by interneurons, as you can see. But not all the interneurons are active at the same time. So basically, if we can adjust how these interneurons work, then we can have a good idea how our new memories are encoded. These cells mainly receive inputs from lower level inputs, basically brainstem and other subcortical structures as well. But why is it important for us? These processes are mainly described in rodents, but at the end of the day, the point of basic research in a lot of cases to understand how humans work and how our memory are generated. To do that, we cannot have any exact experiments conducted on humans. So we have to have raw data that we can use to have a broader look at it. So we try to understand how these different interneurons in the humans are. So we counted them. First, we stained interneurons that are containing parvalbumin. These parvalbumin cells forming basket-like structures around the principal cells, and they can exert very strong inhibition on them. Basically, they can locally manipulate a really huge segment of the principal cells. We found that these have closely the same proportion as somatostatin containing interneurons. These cells are mainly inhibiting the dendritic segments of the principal cells that receive sensory input from the cortex. Basically, they can even encode memories themselves in a funny way. If they can remember how they were activated during an event. Basically, it somewhat resembles what we found in rodents. However, the carotene interneurons are quite funny creatures because they are so-called interneuron-specific interneurons. These interneuron-specific interneurons innervate other inhibitory interneurons. Basically, they coordinate how the interneurons work. And what's quite interesting, that they have a larger proportion in the human hippocampus compared to rodents. Basically, what you can see here is they can be the modulator that can make a distinction between rodents and humans. But we also counted how many GABAergic synapses, inhibitory synapses there are. Here you can see that it's an electron microscope picture. It has all the inhibitory synapses labeled, and we counted them one by one. And also did some reconstructions. Here you can see an axon, a long neurate of the interneuron, of a parabolic containing interneuron. And you can see that the red labeled dots are the synapses themselves. And you're may not familiar with electron microscopic picture of a synapse. That it has some bulge-like structure, so-called button, and on this you can find the synapses. And it's quite interesting to compare these buttons not only in humans and different kind of interneurons, but also to rodents. From this study, we had two main conclusions that I want you to get home with yourselves. The size of the hippocampus does not correlate with the number of interneurons found in it. It is quite interesting in the perspective of the brain, because the brain is not modular on a fundamental level. You can't say if there is more cell, it's more complex. So it works faster if it has more cells. It works fundamentally differently. And the other is in the hippocampus, there is a higher proportion of carotene containing interneurons in humans. Basically, putting carotene containing interneurons in a position that can influence other interneurons and as a whole can influence how our memory generation and memory processes work. With this study, we wanted to build a bridge between rodent and human research. Basically, we wanted to open a new window that allows us to build models based on human data and perhaps understand better how our memory works, how our memory works in different kinds of diseases, and what we can learn just basically looking at the human hippocampus. And with that, I would like to thank you for your kind attention. If you have any questions, please do not hesitate to approach me in the coffee break. Thank you.
Leave.